We'd like to introduce Dr. Jonathan Lundgren as he walks us through the grower report today. And most of you have already received your grower report. And so, yeah, Jonathan, here you go.
All right. Thank you all for tuning in this morning. It looks like we've got a good crowd from all over the place, and that's amazing right off of the bat. Um, so I guess I'd like to start, kind of like zoom out a little bit, right? Why did we do this 1,000 farms? You guys are all participants of the 1,000 farms, right? Which is far beyond 1,000 farms at this point. Why? Why did we visit your farm? Um, and I guess the answer to that is we're in a real pickle right now. I mean, I think you guys can all see from many different aspects that there's a lot of challenges to our food system, be it climate chaos or desertification, water wells are drying up. Just isn't life the same way as there used to be on my farm. Um, or from a socioeconomic standpoint, there are no farmers out there anymore. The rural America is kind of drying up, or really tough to make money in this gig right now. Um, but and so we're all kind of in this same boat together. And at this point, there are farmers that are figuring this out. And so we decided to try to figure out across the country in different food systems, in different regions, what seems to be working and what doesn't seem to be working. And that led us down this path of regenerative, the 1,000 Farms Initiative, essentially trying to sample all of these different places, all of these different food systems, trying to see which approaches are delivering on environmental outcomes, on social outcomes, on human health and nutrition, on economic well-being and resilience. Yeah. So why did we approach it like this? What good is science? What is this bringing to the table? I get asked this by a lot of people, even our own staff, right? I mean, why did we do 1,000 farms? Is Jon just a data monster? He just, I can't get enough of it, right? There's a number of reasons, right? Everybody on this call is trying different things on their own operations. Some of them are working, some of them aren't. Sometimes it's hard to know, right? I mean, we can do these things in our own minds, and we can apply these new approaches, and it's like, yeah, this has got to be working. I feel it that like this was doing a good thing, right? But at the same time, it's really hard to assess that. We have to constantly self-assess. And there needs to be a process to that assessment so that we're not just deluding ourselves, that this is real. What we're seeing out there is actually delivering on these, on our expectations and on our observations. And that's not to discount those observations, right? But rather it's to reinforce them and be able to help us to translate those to other people that have never experienced what's happening on your particular farm. That's where the scientific method kind of comes in. And there's a lot of folks that are making claims right now about what is happening in regenerative agriculture, but very few of them are applying the scientific method. And that's extremely important from an impartial standpoint, no less. Okay? That's extremely important. There's a few central elements to the scientific method that differentiates it from just our own observations. Okay? Replication, data collection, and analysis of that data. Those things are in your grower report. All right. And we're going to be discussing each of those elements here. Replication. Why is that so important? You know how many times I get called by folks that have a demonstration farm or or an experiment farm or something like this. And they're like, boy, we have attained some amazing results. And this is true. I mean, this has got to be true. What these farms often measure is wonderful, and it tells us what the potential of that system is. But all it is telling is what the potential of your farm is. It is not telling us what the potential of farms is. And that's where replication comes in, okay? Measuring across different farming systems whether or not you attain the same results time and again. That gives us a level of precision and accuracy that we can start to hang our hats on as other people are trying to figure out what is the potential of regenerative agriculture or a particular soil health practice or a particular micronutrient application. The next thing is actually collecting data. When you go out on your farm, you're experiencing things. Your senses are taking in information, all kinds of information, from the, the reflectance of the green, plant material to smells of plant volatiles or other things within that system, or insects bouncing off of your skin, or your ears are picking up bird songs, right? All of those elements are data. All right. And we're developing all kinds of gizmos to give us more data, right? Numbers. We're translating all of those experiences and what our senses are measuring into numbers. But that data becomes extremely important because then it's unified, right? And you can compare it amongst different places, amongst different conditions. But the data is only useful without a third component of the scientific method, and that is analysis. And so what analysis is doing is, sure, I went out there and I collected all kinds of data. I got so many more bees on my farm, right? I got so many bees. This has got to be working. But is that number of bees the same as on a conventional farm? Or another farm that's doing something totally different from yours, or whatever? Applying an experimental design or a statistical analysis allows us to differentiate what you're observing and see whether or not that, whatever it is that you're applying on your farm, whether that's producing a result that is truly different from other observations on other places. And so in your grower report, we're giving you your, number one, we went and we sampled multiple places on your farm. We have replication within your farm, but we also measured on 1,700 other farms around the country, other circumstances, so that we could give you that replication. We're presenting your data to you, your actual numbers for many different facets of the system, but then we're also analyzing that for you. And it's all in this automated process that produces, all kinds of little bugs in the system and typos and things like this. But all in all, this is giving you a really powerful view into what's happening on your farm that really doesn't exist anyplace else. So that what your grower report, I hadn't actually looked at it in a long time. And so I was kind of reviewing it before this. I'm like, well, I better speak about it. I gotta, sound somewhat intelligent. And I'm like, this thing is really cool. So maybe that's just, anyways, I'll shut up. Okay. I'm going to share. Should I share? Is there any questions about that? Do we want to open it up at the first, or is there anything before we dive in? I can't see anything. I don't know what I'm doing. So
Yeah.
oh my gosh, there's 37 Q&As.
that. There's lots of things.
Oh, okay.
Yeah, yeah, you see something that you want to take it? Yeah.
Okay. What is the percentage of the Thousand Farm project that is in Canada? We visited two Canadian provinces so far. We visited Saskatchewan and Manitoba, and we visited about 100 farms from up there, so a lot. I wonder if okay, we'll talk about that. Used to, okay, and then, yeah. All right, I'm gonna dive in. What do you think? Well, is that all right with you?
Yep. Sounds good. Yeah.
Okay, I'm gonna share my screen and we're gonna start looking at an actual grower report. Okay, share. Can you guys see this okay? Is it there, Lynette?
Yes, I'm seeing. Uh-huh. Yep.
You're seeing a map?
I am.
Okay.
I'm seeing the map of 1,000 farms. Yep.
Okay, so this is where we have been so far. We actually do have Puerto Rico on this list now. We'll be in Georgia next week, I think, something like that. And so we are continuing to fill in this map. What I want through the end of Thousand Farms is for this map to be a reflection of the nation's food system and ultimately the world's food system. All right. So you kind of get an idea of the different regions and what states and things like that are in each of the regions. Those will become a little bit more important. Just as a side note, Blue Dasher Farm in Ecdysis's headquarters is right there in eastern South Dakota, right on the Minnesota border there. Our field day is August 8th. Come on out. We'd love to see you. All right. So I'm going to talk to you a little bit about these graphs first off. Many of you have had analytics done on your farm, maybe a soil test or something like this, and you're always given like just piles of numbers, right? And it's really hard to know what those numbers mean. And some places are better than others, but a lot of times it's not real transparent exactly how they're interpreting these things. So I want this to be as transparent as I can. Okay, so what you are seeing on here, oh yeah, from each of these farms, I did want to mention that. This grower report is the tip of the iceberg, right? Uh, so we actually collected on your farm, depending on how you cut it, 500 different data types. And all of those data types, and if we are looking for like species level identification of your birds or microbes or invertebrates, that'll all be available via our grower portal, which, as you can imagine, is a monumental task for trying to do that on 2,000 farms. So that we'll have an actual, you guys will be getting that link and stuff like that as this grower portal comes on in Q2 here. So heads up, much more data is coming your way. Okay, so here is an example graph. This is not your farm, but there are, yeah. What do you guys all see on this graph? First off, you see 3 bars. Your farm is the green one, okay? And usually you'll have your site code on there. It doesn't say your name, it has a number. We're going to make it mandatory for participation to have that number tattooed on you someplace going forward. So just heads up on that. No, we're not actually going to do that. Then you have these other 2 bars on your graph, okay? You've got the regenerative and you have the conventional, okay? So this is not what some certification program is saying or what Syngenta decided you're now regenerative or something like this. This is based on a quantitative metric that we have developed in-house and put $1 million worth of data behind. that kind of helps us to distinguish for our own scientific purposes what regenerative agriculture is. And it's really functionally, it's based on outcomes. And you'll see that throughout the data sets that we're generating here, is that regenerative is meant to attain certain broader outcomes. Carbon capture and storage and reducing greenhouse gas emissions. That's a big one. Climate chaos. Regenerative agriculture is meant to increase water cycling and the efficiencies of that, reverse desertification. Okay. It's meant to increase biodiversity, life on your farms in all of its forms. It's meant to increase nutrition of the food products. It's meant to reduce pollution. It's meant to increase the resilience of your farm, economic resilience. And then it's also meant to improve the wellness, physical, mental wellness of the farmers themselves. Those are some of the outcomes that regenerative agriculture is supposed to do. Our scoring matrix or our rubric actually helps to, it actually scales really well with that. We can ask a few yes or no questions about what you're doing on your operation, and it becomes very predictive. Regenerative agriculture is not and cannot be practice-based. If we make it practice-based, we're dead in the water right now, okay? And so that's something that's really important for us to remember and think about as we're discussing regenerative agriculture. The definition as it is applied within your grower report is available. It's a peer-reviewed study. It's free and open to the public. You can take a look. You can use it as much as you'd like to. Yep. Everything that we're doing, we give away for free. And so you see the two different treatments. Those had to be kind of honed in. Because comparing your farm to national averages in all systems, yeah, that's great to a point, but it's also it really muddies the waters, and it makes it very difficult to understand what's going on in terms of context. So that regenerative and conventional columns, those are only comparing your site to a food system that's closely related to yours within your ecoregion. So what you'll see, for example, on this particular example graph is regenerative in California orchards. And honestly, California orchards means California almonds. You also see the level of replication in those other bars. Observations 32, observations 64. That gives you some level of confidence as far as what it is that we're comparing you to. If we're comparing you to something that has 3 observations, that's pretty weak, right? That's pretty weak. You need to have a big difference in yours to be significantly different from that. That's why we replicate so much, is to give us some level of confidence of where you're sitting on your farm and what practices might be able to help you on your farm. Okay, then above each of those columns, you end up seeing a number, and that's the actual value. So that should correspond over on the y-axis of that graph. And then also another thing that's worth pointing out is that you see those little whiskers on each of the bars. Those are your standard errors of the mean, okay? That means variance. That gives you some level of precision, all right? If you have great big error bars on your graph or on one of these other graphs, that means the data is all over the map, okay? If you have a very tight distribution, that means that usually that gives you some level of confidence that if you're using a particular system, that you will experience those same results. Okay, so in this graph, you have replication on your farm as well as replication across the whole region. All right, this is an orchard from California. And these are all anonymized. We never tell people. We will sometimes tell, especially if you're okay with it, We will tell you that other people that you're involved in the study, like we went to your farm, but we will never, ever, ever, ever share your data. Okay. Okay, a bunch of mumbo jumbo that's interesting and you should read. And then we get to the data. All right. Questions right now. Anything, Lynette, on the chat that I need to attend to before I move too much further into the actual numbers? Did I lose everybody? I can't see anybody.
Nope. I think keep on going. You're doing good. Yeah.
Keep on a going.
You're just
All
listening
right.
right now. Yeah.
Okay. So we have a very interesting farm on this one. The first data set that you're going to be looking at right now is of the Haney Soil Health Score. Okay. So normally, if you guys were all sitting in front of me, I would ask you a bunch of questions like, what do you think the Haney says? Why is this so special? And those kinds of things. So I'm just going to have you like put your mental, all of those questions in your brain, and you can ask them later. Work. That's just, yeah, not the way this format works. So we'll try to get to that. Okay. Haney Soil Health. What is this thing? Why do we care about it? Okay, so there's a lot of different physical and chemical metrics of the soil, and those are great. I mean, those governed our soil management decisions for many, 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 many years. Okay, the better living through chemistry. Our perception of the world was through chemical means, right, and the chemical measurements for a long time. We now understand the importance of biology. And the Haney Soil Health Score gives us another kind of approach to that. And so it incorporates some of the physical and chemical natures of your soil, but it also includes also soil respiration within that calculation. And that gives us a little bit of a different perspective on what's going on chemically within your soil. Okay, so Haney Soil Health is, I mean, it's telling you about the physical and chemical properties of the soil, but also the biology. And, generally, if you have a score of around 7, things are okay. But this raises up a really important point. Because so often when we give these grower reports out to folks, they're like, wait a minute, I didn't win. Right? I mean, my numbers aren't the best numbers in every single one of these categories. Why didn't I win? I think that's, you got to kind of couch this, okay? These are numbers. These can help us to guide what's going on out there on your farm. And that's really important. That's valuable to us. But it's not always great to compare your farm to every other single farm out there because there's a lot of other contexts that have to go on there. And so if you don't have the highest number, that doesn't mean that what you're doing is bad, okay? If you have it particularly low, then you might want to be paying attention and figuring out why. These can be little governing tips. How about we put them that way? So yes, a number of things could influence any one of these different data sets. Parent material, so just your context, your situation on your farm, the parent material that's lying underneath your soil, that governs the chemistry of your soil in a lot of ways, and the life, the climate that you're living in, that's going to influence it. The food system that you're producing. If you're raising cash grain, the potential of that system is much different than if you're raising animals on perennial pasture. One of the real benefits of, we were down talking in New Mexico on where we presented some of this in person to the farmers near Taos, and one of the farmers there had a really interesting perspective. She said, I thought what I was doing on my farm was really great. And I was actually worried that I was peaking out, right? Like I was starting to asymptote as far as what the potential was. my numbers were not as high as the farmers in this room, which means that I can continue. I'm really happy with my farm. I can continue growing more and more things. I thought, boy, that's a really healthy attitude, isn't it? So we can all strive to improve. And we have not seen asymptotes in a lot of these different data sets within regenerative systems. So that gives me a lot of hope that what we can continue to do is grow things on our farms. Another important aspect of this is sometimes the best way to use these numbers is to think about how you're measuring your field over time, maybe over the season, but especially across years. So if you're implementing something new, a new practice or something like that, getting additional information over a long period of time can start to give you a picture of whether or not what's happening on that farm is working, whether it's delivering on some of these promises and outcomes that a farm is supposed to deliver on. Okay. So that's Haney's and much more than Haney's. Yeah. I'm gonna buzz through a lot of these things and then we can ask specific questions. Does that seem reasonable? I don't know. What do you want to do, Lynette?
That sounds great. Yeah. And just want you to repeat one more time, what's the number in parentheses next to your site mean or stand for? You know, the site that you want everybody to have tattooed on them.
Yes, your tattoo is your site code. And that one's really important because what that does is it identifies you to the government. No, not you do not give any of your information. Yeah. What that does is allows us to anonymize all of your information. And then it also allows us to link data sets because we're collecting a lot of different data types on your farm and they're in different spreadsheets and things like that. That becomes our unifying number. So it's more for us internally than anything else, but you will see that your name is not on this report. And I mean, that's because we take your data so seriously. We do not give that away. All right. Did I do my job there, Lynette?
You did. Perfect. She said, okay. Thank you.
All right. Organic matter. Organic matter, that's carbon, right? If we're to make this as simple as we can, that's carbon. That's the breakdown of plant and animal material into your soil. All right. That is the productivity of your soil in a lot of cases. All right. That governs the potential things like water holding capacity of your soil. So how much that sponge can hold. That is also a good reflection of how much fertility is in your soil. We're finding that this one is a really good metric of a lot of different things. Organic matter. So if you're increasing your organic matter, so you're seeing those different numbers, right? I mean, you see the regenerative almonds and you see the conventional almonds there. And the conventional has quite a, it has, 2.8 versus 3.3% carbon or organic matter. But that particular orchard this one has 5%. You know how much what you guys are showing us, what you farmers out there are showing us is that soil carbon can grow much faster than science ever thought was possible. We're seeing farmers improving their lot really darn quickly across the country. That gives us a lot of hope in terms of using our food system for combating things like greenhouse gases in the atmosphere, because we can take that carbon out and put it down into our soil and make it start working for us. Yeah, okay. So that's organic matter. All right, now we're into phosphorus. And I could even, I could even show some other different systems just in case you wanted to know. Like, okay, this is croplands so this is an example of croplands that they had converted over to rangelands. This is a project that a grad student in our group is just getting started on. where Cass Cool is she is studying how these systems change and how quickly. But man, I mean, when you start looking at it, the carbon levels increase. I mean, they start jumping up even in year one. It's nuts how fast this thing changes. Look at the Haney test compared to other. I think that these must be. I don't know what a NAN is, but that's one of these little snafus of the automation process. But essentially, yeah, you can change your system pretty darn quickly by going into a perennial system, putting animals out there. Okay, phosphorus. Look at this one. This is really weird, huh? And this happens a lot of times with different micronutrients. So you'll see other micronutrients in your soil down here, and we'll be giving you a lot more of these in the grower portal. Wait a minute. I thought regenerative agriculture was supposed to only increase things, right? Why on earth do we have such high levels of phosphorus in these conventional southern sites? Well, a lot of that, there's only a certain proportion of that phosphorus that's bioavailable to plants, the inorganic forms. And so when you fertilize, what ends up happening is that you smother that environment in this plant available nutrient to really jazz your plants up, right? But what that does is it displaces the microbial cycle that does that for free, which normally takes in organic forms of phosphorus, things like plants and animals, and turns them into plant available nutrients. That may act a little bit more slowly, but in a healthy system, is way more effective to the longevity of your field or pasture. And so interpreting those different micronutrients, especially when you're getting high levels of things like inorganic nitrogen or inorganic phosphorus within conventional systems, that's not necessarily a good thing. And so, yeah, we're presenting the numbers. Sometimes it's up to everybody to try to understand what those numbers are. And that's a big part of our job right now with 1,000 Farms. Our scientific teams right now are analyzing all kinds of different aspects of this and trying to prepare it for the peer-reviewed literature. Okay, this is a silvopasture. That one's pretty interesting. Yeah, I don't know how much we want to dig into each and every one of this. Bulk density is basically how fluffy your soil is. Organic matter really has a big effect on that. The soil pH, that's an important one. It's also related to calcium, magnesium levels within your soil and parent material. But one of the interesting aspects of this is that a lot of times within the scientific literature and soil scientists, sometimes will make the claim that, this is some of these factors, especially micronutrient levels, are strictly related to parent material or the addition of inputs. And what we're showing as part of 1,000 Farms is that things like inorganic carbon or calcium or magnesium tend to be very high on regenerative operations without the inputs that we're getting. So that's what we in the business say biogenesis, right? Rather than lithogenesis. And so what it does is it tells us an awful lot about what the opportunities are with regenerative agriculture to correct certain soil imbalances in some of these micronutrients. Okay, I'm just going to scoot along here so that we have plenty of time for questions because I see that I have already run my mouth off. So in each of these, we have kind of talked about bulk density, pH, organic matter, all of these sorts of things. And so you can kind of see some of at least the initial conclusions as far as what they mean and why these things matter. Now we get into the next topic area, and that is water. So we present in your graphs, you get two different measures. And we should be presenting water conductivity as a log transformation because right now it looks like everybody just has zero, but there's obviously not zero. It's just a very, very, very small number. So by turning it into a log transformation, we can tighten up those residuals and you can get a little better picture of comparatively. So bug number 1,752 that we have to try to address on these, on everything in our lives right now. Soil conductivity. So what you're seeing there is from an infiltrometer. It's a mini disc infiltrometer. We find that the mini disc infiltrometer tends to scale better with regenerative outcomes than the infiltration test, the ring test. A lot of times that's because the ring test is influenced by so many other things that really haven't all been teased out yet. Things like, the percentage sand in your soil or organic matter in your soil, or whether there was just a rainfall event or whatever, right? All kinds of different things. The conductivity is measuring not just the gravity pull, but rather pore size and other aspects that are kind of dragging that water and allowing it to permeate into your soil. You want high numbers. High numbers is good. And after doing literally tens of thousands of water infiltration rings, what I can tell you from this is high numbers are good, but low numbers are not necessarily bad. And so interpret the water infiltration data a little bit carefully. Don't, jump off a building if you don't have the highest number in that one, okay? In fact, don't jump off a building no matter what numbers we give you. The
Hey, John.
water infiltration, yeah.
Yeah, Scott's wondering that if his infiltration is at 47, he's not sure of the units. Is it 8 times the national average?
That is milliliters per second that are infiltrating into the water or into the soil. Sorry, a number of other folks use inches and stuff like this. There's all kinds of data in the scientific literature that is really tough to interpret on water infiltration rates because people don't use the scientific units. And they're not always characterized by the volume of water that's being put in. They're put in by inches, which is like actually that's not a volume, it's a distance, right? And so it's a little bit tough to interpret. And then the amount per time is also really important. So if you've got 47, dear God, I mean that's like when you flush the toilet and it's like going around, you've got like a centrifugal, that thing's going in pretty fast, right? That's amazing infiltration rate.
And so he's wondering, is it due to management or soils or something else going on?
Great question. Probably both. You probably have a soil that's ready to receive that water, but then at the same time, your management will influence that. So things like making pores in the soil, like little channels for that water, that's life right there. That's decaying roots, that's earthworms, things like that are punching holes in there. Okay, so that is water. You now are experts at water. You want water, but not too much. All right, now we get into my favorite part. And one of the things about Thousand Farms that makes our study pretty dang unique from anything else that's out there is biodiversity. We are measuring life on a scale that I dare say hasn't happened before. This is the third largest bioinventory that humans have ever created. We're behind La Selva down in Costa Rica and the Smoky Mountain bioinventory. They each had like 20 years. We accumulated species records of 15,000 taxa within 4 years with a minimum of like 30 people working in a barn in the middle of nowhere, South Dakota. It's pretty cool. It's pretty cool. And what we're discovering about that is that all of the regenerative outcomes that I listed before, be it uh, carbon capture and storage, nutrition of the food, farmer happiness, economic resilience, water relationships. All of those are outcomes of one thing: growing life on your farm. Every decision that you make should ask yourself, is this growing more life or killing life on my farm? If it's killing life on your farm, do something else, because you are never going to reach the potential of what your farm is capable of unless you change that mindset. Okay. So what we see here, plant biomass, this is what we measured with the plant drop plate meter. It's a compression plate that squishes the vegetation down. So you see that on regenerative, or in this particular silvopasture, they've got a lot of plant material. We also scanned the soil surface within a quadrat, and we measured the proportion of green cover on that. Okay, so how much green material was shielding the soil. And then we counted how many species were on that within that quadrat as well. So you get an idea of how much plant diversity is on your fields. You may recall this orchard, who is really wonderful and a world leader in many things. And what you see is the plant in the middle of the Central Valley of California is twice more than the conventional counterparts. And this is what we're seeing within these orchards is, man, the way that they're making these orchards function is by growing healthy plant communities. So they also have great ground cover. That means that a lot of their ground surface is covered by plants. That's super important, okay? But in this case, take a look at this. You end up seeing the number of plant species kind of drop down. That doesn't mean that there isn't like a lot of diversity in terms of plant communities out there. What that means is that, there might be a little bit of an imbalance within the community where you have a number of species that are really well adapted for that particular farm, and they're able to produce a lot of biomass and kind of take over that niche. So it's a little bit more of a simple community that way, but that doesn't necessarily mean that it's a bad thing. It may be that plant community is starting to heal itself. It may be an artifact of where we're sampling. I happen to know that this particular farm is transitioning, that they are using and integrating sheep into their operation, and maybe the sheep are selectively consuming certain plant species, at least at this stage, until that plant community heals from that change. And so it just kind of depends on where things are at. This is a potato field. And we're comparing it to row crops, which isn't a perfect comparison, because potatoes are just really hard on the soil and on life in general on farms. We haven't seen great potato fields, but this one is particular. This one's probably the best one in the country. I'm going to know this one. And what you see is that they are able, their numbers are maybe 60% ground cover maximum. I mean, this is better than anybody else that's out there. And you can see what happens on, there's zero other plants in conventional systems. And that's, for row cropping systems, that's pretty typical, right? You end up seeing that. But boy, look at even within this particular farmer's own operation, you see a lot of variability. Isn't that interesting? Okay, so plants are good. They're taking that carbon out of the atmosphere, they're putting it back down into the ground, and the biology, the diversity and the abundance of dang near every other organismal group in your system scales with these metrics. How many plants you have within your system. Okay, super important. Then normally we would go into, okay, so here we see microbes next. All right. Almost always what you see is in the regenerative column, you see way more life. And if you're transitioning your farm, another pattern that we're seeing on a national level is that you end up seeing um, this is the first response that you end up seeing. Life returns to your farm, and then that life does things. Okay, it grabs carbon, it punches holes in the soil, it makes more organic matter, it makes you happier. It provides resistance against pests and things like this, and reduces your dependency on agrochemicals. And so these numbers generally are always higher in the regenerative, especially for microbes. And so you have fungi, you have bacteria, total microbial biomass, and then you have these ratios of fungi to bacteria. Okay. The grower portal is going to reveal a lot more information about these different, yes, what should be expected under different scenarios there. So,
Uh,
okay, and then yes, yes,
Question
hello.
on that grow portal. One farmer wants to know, are they going to be able to see what other farmers are doing in
Yeah,
their area? Are they going to be able to
yeah,
see that data and be able to compare it? Will they be able
yeah.
to? Yeah.
That's the whole idea. And as Avalanche comes on, we're actually putting the keys of the kingdom into your hands. So you can sample your own operations and then upload your own data into the portal. What it does is then it relies on this sort of citizen engagement, right? Where the contextualization then becomes like, okay, this is regenerative and conventional production in your area compared to your numbers on 500 different data options. And then it also gives the option, if you select, to be able to, join groups with other people and stuff like that that can answer questions like, hey, I need to figure this cover crop thing out. It seems like the data really suggests that it's working for some farmers, and I haven't been able to figure it out, but it seems like these other folks are. So I need to talk to somebody. That level of support really is important right now. So we're excited about the grower portal. And now we would jump right into insects, but you didn't get that information. But so we're going to bypass bugs in light of birds. And birds are a really good one too. This is the number of birds that we saw and the number of bird species that we saw. So if you had a big flock of starlings blowing through, then you might artificially inflate abundance, but species richness would not be affected by that. Okay. So you get this idea that potato growers are kind of, yeah, not seeing the same birds as other places. And now we see silvopasture, and I'm scrolling. Okay. Yeah, that's only regenerative, so you never really get the idea. And here is some rangelands from North Carolina. Might see something good here. All right. No, you do not, because these rangelands tend to be a little bit more stable in this area. Okay, well, you get the idea. So, one of the things about birds is we do end up seeing a 30% increase within regenerative systems on a national average and often within individual clusters. Um, but birds are one of these things where they move a lot. And so sometimes it takes a little bit longer for them to return to a habitat in measurable numbers. That doesn't mean that they're not super important, okay? And they are. In terms of grower engagement, especially, farmers love birds. And so that's one of the reasons why we've been so good about measuring them. We also, this does not incorporate, this just has data from our own ornithologist visiting your site and doing a walkabout. Okay, we also put out data recorders where we're measuring bird songs on your farm and identifying the community that way. We're just getting Syrinx up and running on that. So that's our AI algorithm that should be able to identify all of the birds on your farm. So stand by for even more of that sort of information. Okay. So in a nutshell, that is Oh yeah, why didn't you get bugs? If you're from 2025, why didn't you get bugs? I have a small, yes, we have this. This is why. We'll stop it there. Essentially, we're going through your samples right now. We've developed an AI algorithm that can identify insects. It's really the first of its kind. We're leading the entire planet in terms of developing this image recognition from a research standpoint, and it's about 85% accurate. We're now trying to figure out how to get it to recognize multiple specimens per image so that we don't have to individually record every single bug kind of a thing, which that video would have illustrated really well. But until that new version is up and running, which will probably be another month or two, we're going to start rolling out the insect data from last year, stepwise process. So that is where your bugs are. Grower Portal is going to have more bugs than you can shake a stick at. Not that you would ever want to do that.
Okay. And will the grower report updated once the bug data has been entered? Or how will that work?
The portal should take it over at that point, is my hope. Okay. Unless, I mean, if people need a printed copy, that's fine. I guess we could try to figure that out. But okay. Hoping that the portal kind of starts to take over the old grower reports.
And so we have a question here on high infiltration but low soil conductivity. What does that mean?
Yeah, that kind of stinks.
That a lot in reports.
That's the big problem, right? Is that the hydraulic conductivity of the soil seems to scale really well with regenerative outcomes, and the infiltration ring data is the one that everybody does, but it's all over the map as far as what it. So what does it mean? We're going to be trying to figure out what the cofactors are behind interpreting the ring data over the next little while here, and hoping that we can start to interpret those data a little bit better and explain what those mean. That should unlock the ring test for a lot more usefulness moving forward. For right now, think of the ring test as a great opportunity to kind of see how your soil changes over time.
So you'd still say use the rings because they're most readily available.
Yeah, it's super easy and stuff like that, but don't compare your ring test to other people's ring tests.
Yeah, just see what changes on your farm.
Yeah. I mean, you'll see numbers compared to other places, and that could inform you going into understanding what's going on your farm. It doesn't mean it's a bad thing. Just don't get upset if you don't have high infiltration reference or in reference to other people.
Yeah. And soil infiltration, does the time of the year matter? Late summer versus early summer?
Oh, for sure. No, yeah, absolutely. Add that to the list, right? Of all of the things that could affect infiltration rates. Biology is a huge one. Management, tillage, all of those things are going to affect it. And so it's a hairball. We're going to try to tease it out. We've got a lot of different data that we can kind of try to help interpret that ring test a little bit.
Yeah, so it's really not looking at one thing, right? It's looking at all the different data points, right? Looking at the full picture.
Absolutely. It is about the system, isn't it?
Yeah. Yeah. And just to see in the changes, right? And being able to sample it yourself. We have a Canadian that's asking, we don't use this Haney soil test. It's not available by any testing facility in Canada. Are you familiar with the differences in the testing procedures?
Differences in testing procedures like other tests? Yeah, there's a whole bunch of them. And every lab that you send your soil sample to does their methods differently. And so even if you've got, like, if you're measuring the Haney test at a couple of different labs, you might get totally different results. And that's why we've had to kind of marry ourselves to one particular facility in terms of just replication of this study and cross comparability. So something to bear in mind, choose wisely. Oh, if you're getting soil tests done on your farm, send a few dummy tests in. That's what we always do in order to double check. Did you get the same numbers when you send them the same sample? And if you send it to other places, if you get completely different results, then yeah, figure out why. Because don't just trust it because chemistry is inviolable, right? No. There's a lot more to go that goes into that.
So really, it's
Uh,
consistency, right? To sample at the same time of the year and to sample also,
oh, for
or
sure.
sending your sample right to the same lab or the same type of of, uh, of sampling, right? Or
Yeah,
of,
for sure, as you
yeah.
said. Mhm.
Mm-hmm. And so when you're talking about organic matter, does it mean more biodiversity at the soil level?
Yeah. Yeah, essentially that it scales almost, yeah, very directly with almost every single organismal group that we measured. Scales really well with organic matter. And it may be a chicken and egg kind of a thing there. Did the organic matter become as a result of the life on the farm, or did the life on the farm come because the organic matter was higher? I think that based on what we're seeing at this point, it's circular, right? I mean, this is a feedback mechanism within your farm. More organic matter gives more life, which gives more organic matter, which gives more life. It's jump started by getting life on your farm first. So getting plants out there. And that doesn't mean buy a jug of bugs, right? And pour it on your field. Because if you do that, all you're doing is if your field is not able to receive such biodiversity, then you're just, yeah. Um, yes, you're not going to attain the results that you're looking for.
Yeah. They have to have an environment, right, for to grow and flourish, right? So that
Right.
environment has to be there for them.
Plants and animals are your biggest tools. That is, oh yeah. I mean, so why don't you talk about what we have to do for each of these different chemical and physical tests? Because it's only one answer. I mean, this is what we're finding time and again. It addresses many of the deficiencies on your farm. Perenniality, plant diversity, keeping the ground covered all the time. These are practices that contribute to these things, right? Animal integration and croplands and diversity in terms of revenue streams and heritage varieties versus conventional hybrids. These are elements that universally address many of these outcomes.
Mhm. And so when you're looking at pastures, why would irrigated pasture be compared to an arid rangeland? Why would you include those two in the same comparison?
I'm assuming that that's what was done, huh?
It must have been, yeah, which
Hmm.
is a possibility, right? How does irrigation affect, pastures and
Oh,
compared to some?
sure. Okay.
Yeah.
Yeah. So taking water out of the ground and putting it on the soil surface and stuff, that's going to get you maximal probably within arid environments. It'll probably really make your croplands flourish while you've still got the water. But that's starting to go away. And so in some ways, it's kind of giving you a comparison of best case scenario for crops versus a perennial rangeland. So what could you do on your croplands versus making it go perennial?
And it's interesting too. When we were looking at irrigated pastures, it seems like, yeah, certain plants really like water, right? They're the ones that have to be watered all the time, and it's usually those planted crops. While an arid rangeland really does like when there's water, but also when it's dry. It seems like
Yeah.
different plants flourish at different times of the year.
Yeah. And if you don't get rainfall, I mean, start thinking outside of the box a little bit. Stop trying to put a square peg into a round hole with crops grown in an area that there isn't water. Um,
Yeah.
you're paddling upstream, and we're finding across much of Western North America at this point um, that farmers are starting to transition those prime crop fields in arid areas um, back to perennial pastures, and they're doing it very successfully and making it work. And I think that that is an important component of the future of our food system.
We have a question about what lab do you use, what to send your soil samples to?
Where did we send our soil samples? So we are not married to anybody. Um, we send our samples to Regen Ag Labs for chemical analysis, Haney tests, PLFAs. Um, we found them to be very, yeah, they do a good job, and they're at a good price point for what we're trying to do. Biome makers is how we sequence our nutrient, or I'm sorry, our microbial communities for eDNA. We have been using Edacious as part of the nutrient assessments that we've gathered. And then for insects, birds, plants, and a number of water holding capacities and things like that, we do that in-house in our own laboratory. And then the social and economic stuff we do in-house.
And so we have a question on rangeland. He says my phosphorus is 10, regenerative northern Rockies and plains is 39, and conventional is 36. What's a good number? Do I need to address this?
Are you happy with the way that the plants are being produced right now? What are ways of increasing that? Regenerative style grazing, keeping animals tight, letting it rest, grazing it for a short period, that's going to be really important. If you're in the western half of the country, we're finding that the longer rest you can give it, the better. And so certainly one graze a year is probably the maximum that you'd want to do. So take a look at your grazing practices and see if that might be contributing. But again, just because you're lower than other places that we've measured doesn't, I mean, my goodness, especially in the arid West, you can be in a pocket that just has lower phosphorus levels than other places.
For a farmer to start transitioning, what's the first step that you like to see?
Adopt a full system on a small scale. So don't bet the whole farm on it. But what we've seen is we've actually done some preliminary analysis on this. The two general strategies would be I'm going to dip a toe in, I'm going to adopt no-till this year on my cornfields or something like this, and versus I'm going to grow heritage corn on no-till cover cropped polyculture, grazing it, or something along those lines. And what we see is the folks that most often fail are the ones that end up dipping a toe in rather than a full system. Because that adopting of a full system necessitates a mind shift that seems to underscore the attainment of maximal outcomes. But I totally get it, right? I got, 2,000 acres. You want me to do it on? No, I'm not saying that. I'm saying try developing a system on 40 acres, or 10 acres. I don't know what size your farm is. But try and work on that system, and then you can scale it up, right? Because I think if you do it that way, you're going to get hooked, and it makes farming fun again.
Another question is, how is the information gathered, used by your company? And what is your long-term goal for this project?
We're going to change agriculture on a global scale. That is our goal. That is a longer term goal. We're going to start one continent at a time. That's why we started here in North America. Okay. What were the other elements of that question? But we're doing it by supporting farmers that want to change. And so giving them the tools to see whether or not what they're doing is helping, as well as resources in terms of support and social connections that can allow them to ask questions and know that there's somebody else out there that actually cares what they're doing. And maybe has even walked in their shoes a few times. Does that answer it? I don't know if I got every element of it.
I think so. I think that's good. Yeah. So another question on the results. So when you are calibrating the results to compare our results with similar farms,
Yeah.
they're wondering if there's how you do that. How do you choose which farms that you're going to compare, their
Yeah.
farm to?
So we categorize things, right? And that's imperfect. Okay. So yeah, that's one of the, so this is not a peer-reviewed study. All right. We're doing the best we can with minimal resources in order to get you as much information as we can. And for peer-reviewed, I mean, so take corn, for example. Right now, if you're growing corn, then we are comparing you with other cornfields from your particular region, which we've kind of designated where those regions are on the map on your grower report. Okay. If you're growing bloody butcher corn and we're comparing you to DeKalb, 70-30 or something like this, right? I mean, those are different mindsets. Those are different goals of that. There should be way different expectations from those two systems, right? But the nuances of that get to be so extreme that it becomes very challenging. So what we've done is when you see, okay, it's rangelands from from the upper Midwest or something like that, then those rangelands might be grazed by cattle, they might be grazed by sheep, they might be grazed by other things. But we do try to categorize things to kind of help make sure doing our best to make the comparison as meaningful as we can in short order.
A couple more questions on the grower portal. When will it be available, and will they be able to see their raw data?
Yes. They will get to see as deep a level as they want. Species level identification of microbes, species level identification of insects. And we're hoping that there will be fact sheets on as many of these things as we can get. So the fact sheets are being written as we speak, actually edited as we speak. The grower portal is functional. We're incorporating a lot more of the biodiversity information into it now, and it should be accessible within the next few months. And then we'll figure out all of the ways that we screwed it up.
And what about nutrient density? If we sent in either meat
that'll
or
be there too.
Fruit,
yeah,
will that all be available in there?
Yeah.
Mm-hmm. And have we got any updates on that for the farmers as to when?
It's been really slow. Okay. So we've gotten some information on some of the samples. It's expensive and it's slow. And so in part because of where we're at in this process, there just hasn't been a real cost-effective nutrient analysis facility. If you guys know of any, let me know. But we've been using, so we're kind of on the front edge of this and working out some bugs out of that system in order to make it kind of work stream a little better. The other part of it is we couldn't afford to do it all. And so we've got we've got meat, milk, some of the cash grains, almonds. We've got, I think, nuts. So hazels we've probably got. But a lot of the vegetables, I don't know that we'll have the cash to do it. We've got the samples; they're ready to be analyzed if I win the lottery.
Yeah. Right. Let's see if I missed anything else. If I missed your question, go ahead and put it back in if I didn't cover it. We did have one question about reports missing some information. Data points that were taken, they weren't on the report. And this would have been
Yeah.
Arizona.
Yeah, there's a whole bunch. They'll be in the grower portal. We had to choose from, which of the data points can we put into this 30-page report out of the 500 data types that we gathered. So we're hoping to, yeah, give all of that information. If there is something specific that you want, holler at us. Lynette, holler at Lynette.
Yep, yep. Just let me know. Yeah, and I'll take a look and see. Let's make sure I've covered everything. Yeah, I think so. Yeah, I think just yeah, I think one of the things that we're definitely going to talk about is how can we make it so that everybody can see each other as part of these
Yeah.
webinars and to be able to talk about each other. Yeah.
Yeah, that'd
Oh,
be a
we do have
fun.
a question about how do you store the vegetables that are awaiting nutrient density testing?
You freeze dry them. So we call them lyophilize. So we have to dry them down. And so the nutrition, they end up being shelf-stable that way. So that's nice. But not rat stable.
Yeah. Yeah. The battle with the rats, right? Yeah, exactly. Exactly. All right. I think that's everything. I think we covered all the questions, unless somebody else, if I missed something, let me know. But I think we got it all covered. Well, thank you, Jonathan. I so appreciate you walking us through this. And yeah, and like you
Well,
said, if you have any questions, just, yeah, shoot me an email. Most of you have my email, lynette.miller@igdices.bio. So, yeah.
just to leave on, I think it's worth reminding the farmers just, I mean, you guys are the ones who are showing us what's possible. All right. None of this is, we aren't telling anybody what to do. All we're doing is measuring different things and telling you guys and gals what we're finding out there. Um, so thank you for pushing the needle, for trying new things, because until you do that, we will not know what is possible within your food system and your regions. So yeah, boy, Thousand Farms is only possible and only meaningful because of your bravery.
Yeah, and when we're in the field, a lot of times people will say, well, I don't think that's possible. And we'll be able to say, we've seen it. And that's been
Yep.
the amazing thing about, the 1,700, plus farms that we've been on, is that we've seen so many different food systems and we've seen what's possible in each one of them. And it has been an amazing experience. And truly, it is that belief of that farmer that it can be done, and they do it.
Yeah. Yep.
Yeah,
Yep. That's right. Somebody needs to
exactly.
just do it. All seems impossible until somebody does it.
That's right. That's right. Exactly. Well, thank you very much. And yeah, until we meet again, our next webinar will be with Lance Gunderson as we go through the PLFA test. And I'll make sure I get those sent out to you before it. And yeah, it'll be May the 6th is when we were planning it. So.
And then we also have a webinar on neonics next
Yeah.
Friday, not this
Yeah.
Friday, but the next Friday,
The
where we'll
24th
be. Yeah,
of
so that one,
April.
hopefully you guys are all getting emails about that. And then our field day is going to be August 8th. It's the second Saturday in August. I believe that's the 8th. And we have
It's early
burros are
this.
coming out from
Yeah.
California, and Wild Idea Buffalo,
Yeah.
our bison ranch, are
Oh,
coming. That'll be really
cool.
amazing time. We're going to talk about how things change, right? It's our 10th anniversary, so how have we changed? But then also, how do farms change, and what they um, and carry on throughout all of the adversities of life.
Amazing. All right. Thank you all for joining us. Until next time. Goodbye.